Okay, so now the next topic is the internal morphology of the stem of Cycas plant. Now, this is the figure of showing the diagrammatic representation of a TS section of a young stem, okay, before secondary growth, because secondary growth it even occurs in the stem as well, just like in the root. So here first we will discuss about the internal structure of the young stem, that is before secondary growth. Now the transverse section of a young stem of Cycas, as you can see it clearly here in this figure, it shows an irregular outline. Why irregular outline? It is because due to the presence of those persistent armor leaf bases. So these are nothing but they are leaf bases. Okay, leaf bases of both the foliage leaf and the scaly leaves. That is why the outline is irregular and is not smooth. And the structure of the stem, it shows these structures. Okay, first here what you can see just below these leaf trays or leaf bases, you can see the outermost layer, it is the epidermis, and this epidermis it appears irregular, and also it ruptures due to the presence of these leaf bases here. Alright, so the first layer it is the epidermis and it is ruptured due to the presence of the leaf bases, and just below the epidermis you can see the next layer here. This is the cortex. Now as you can see here clearly that the cortex is broad, and the cortex consists of thin-walled parenchymatous cells which is filled with several starch grains. So you can see these are nothing here but they are the starch grains as well as the mucilage duct. So the cortex, it is filled with starch grain and is traversed by many simple and girdled leaf traces. Okay, these are known as the direct trace and these are known as the girdle trace. Okay, so the cortex, it is traversed by these simple and girdled leaf traces, and you can see numerous mucilage canals which are scattered in the throughout the cortex region. And also the cortex, it contains a few cells a few cells of the cortex, it contains the crystals of calcium oxalate. Okay, so calcium oxalate is present in the cortex region of the stem. And just below the cortex here you can see this is the layer of the endodermis as well as pericycle, but the endodermis and pericycle, they are not distinct as in the case of the roots. Okay, the endodermis is not distinct. And okay, these calcium oxalates, they are nothing but they are the biominerals. Okay, the crystals of these biominerals are present in the cortex, and the endodermis and pericycle is not distinct as in the case of the root. And after the pericycle, we know it is followed by the vascular cylinder or vascular tissue. Now as you can see here in this case, the vascular tissue is the stele here, it is eustelic. Why? Because as you can see that the vascular bundles, they are arranged in more than in one or more rings around this central pith. This is the pith here, you can see the pith, and these are the vascular bundles and they are arranged in rings. That is why it is known as eustelic. But sometimes it is ectofloyd polyfascicular siphonostele. Alright, ectofloyd meaning having phloem which is present external to the xylem. So you can see these black in color here, they are the xylem, and external to the xylem, these transparent color is the phloem. So the phloem is present outside the xylem, external to the xylem. So it is known as ectofloid siphonostele. That is a stele which consists of a core of pith, alright, surrounded by the concentric rings or concentric layers of the vascular bundles. In the secondary growth, you can see this is the first layer, second layer, and the third layer. Alright, so that is why they are known as these type of vascular bundles. They are known as ectofloid polyfascicular siphonostele. Alright, why? Because they are surrounded by concentric layers of xylem and phloem. But this is the case of the young stem. That is why only a single layer is present, only a single ring, a ring of vascular bundles. And the vascular bundles here, as you can see, they are narrow and also radially elongated, conjoined. Why? Because they are joined together. You can see this is the xylem, this is the phloem. This is the xylem and this is the phloem. So the xylem and phloem, they are conjoined, and also they are collateral. Why collateral? As you can see here, these black in color here, that is the xylem, they are present towards the pith, whereas the phloem, they are present towards the periphery. So that is referred to as collateral, meaning the xylem is towards the inner side and the phloem towards the outside. They are also open. Why? Because here you can see the presence of cambium. If cambium is present, then it is known as open. But if cambium is absent, then it is known as close. And also endarch. Endarch because the protoxylem is directed towards the center of the pith, and a newly formed xylem, that is metaxylem, it will form towards the periphery. Okay, when the secondary growth occur in the stem, the xylem, the protoxylem will be directed towards the pith and the metaxylem will be directed towards the periphery. That is known as endarch. And all these vascular bundles here, in between the vascular bundles, these spaces here, they are separated with each other. Alright, so in these spaces here, the medullary rays developed. Okay, so these vascular bundles, they are separated with each other by broad parenchymatous medullary rays which communicate the cortex with the pith. Okay, so here in these spaces, the medullary rays are present which connect the cortex here and the pith. And the primary xylem, it consists of both tracheids and parenchyma, but the vessels are absent. Okay, that is the water conducting tissue which is found in the xylem, they are absent. So the vessels are absent, and the protoxylem tracheids, they possess loose spiral thickenings, whereas the newly formed xylem, that is metaxylem, it possesses tracheids. The metaxylem tracheids, it possess the scalariform thickenings. Alright, so just remember that the primary xylem, it consists of the spiral thickenings, whereas the newly formed xylem, that is metaxylem, it possesses scalariform thickenings. And also you see here that the xylem, in between the xylem and the phloem, there is a cambium, right? There is a cambium. So the fascicular cambium, this is known as a fascicular cambium. It is present in between the primary xylem and the primary phloem. Alright, here the primary xylem and the primary phloem, here in this case it is referred to as the vascular cambium. And this vascular cambium is present in the form of short-lived thin strips. And the primary phloem, it consists of the sieve tubes as well as the parenchyma. That is the sieve tubes, the specialized cells, and also the specialized cells of the phloem, and also it consists of parenchyma. Here in the phloem, the companion cells are absent. That is, the specialized parenchyma cells in the phloem tissue are absent, and this phloem, it possesses a few other specialized cells known as albuminous cells. Okay, the albuminous cells which are physiologically and anatomically associated with the sieve cells in the phloem. Alright, so a phloem, just remember, it possesses a few albuminous cells which is physiologically and anatomically associated with the sieve tubes. And here, as you can see clearly, that the pith is large, unlike in the root of Cycas. Here the pith is large and parenchymatous, and the cells they are filled with starch grains, and also there are many mucilage canals scattered in the pith. Alright, so this is the internal morphology of a young shoot. So in the next video, we will take a look at the leaf traces. Alright, these leaf traces here, which are present in the cortex, and also the secondary growth in the stem.